Thank you all for coming, especially after coffee. So everyone's kind of a little bit jittery. But also we have the person who did the work. Matt Carrick is here. He's also recruiting for his own lab at the University of Montreal in Lyon, by the way. So I can always turf the harder questions to him. Ken left, so nobody's going to ask me about human interpretability, but I'll do my best. Okay, so like most projects in the lab, this was also sparked by some kind of a weird biological observation in a strange animal. Well, it's not that strange, but here, hold on, let me see if I can pull it. Yeah, okay. So this was an experiment done by Aaron Andelman, who was a postdoc in Carl Dijsselhoff's lab, and he developed a persistent stress paradigm for larval zebrafish. Why larval zebrafish will become apparent in a second, but it's got about 100,000 neurons. It's connectome. Depending on your definition of what kind of modality of connectome, we know a few things about it. And so the experiment involved head-fixing larval zebrafish and shocking them with 1.3 milliamp shocks, open loop. So the fish aren't going anywhere, no matter how hard they wiggle their tails. And then there's these two distinct behaviors on either side of the behavioral challenge. This kind of persistent stress paradigms have been applied in the context of depression-related work or learned helplessness or passive coping in mice, for example, rats also, and has been shown to have some links with the human condition, but at this point, it's very ambiguous. So when the shocks first come on, the animal responds by vigorously trying to evade it and trying to get away. But it can't get away because it's head fixed, but also it's open loop, as I said. And so then eventually, after about 15 minutes or so of this kind of shocking, we think that the animal is experiencing this as persistent and inescapable, and so lapses into this other mode where it's not struggling anymore, where it gives up all movement, and this is called passive coping. So active coping and passive coping are both adaptive. I don't mean to imply that passive coping is maladaptive. Even people have the same thing, right? Like if you have persistent and inescapable stress, people eventually stop struggling. However, in depression and conditions like that, it's thought that the onset point of passive coping is a bit advanced compared to the neurotypical. So again, at this point, the connection to the human condition, very spiritual and level. But anyway, we wanted to understand what was the brain-wide mechanism of this kind of behavioral transition that occurs over a long time scale. And this is sort of the behavior quantified. So what Matt did here was to calculate the speed of these fish wiggles as a function of time. This should say seconds, sorry, went away under the box. And then the little pink bar that you see here is the duration of the behavioral challenge. So over a very long, this is a very kind of long experiment. And so you see two different traces here. In black is control fish. This is the tail wiggles of 5 control fish, average worth of fish. And then in blue is the shocked fish. And so you can kind of see both of those phases, as well as how squishy this phase transition is. So for people that have been trained in physical sciences or engineering, we want to see a phase transition as a function of an order parameter, right? There's a jump. Biology is gnarly and weird, and so then this phase transition had to be squishy and kind of slow like this. So the two phases are active coping, where the shocked animal responds by elevating its kind of evasive behavior, and then gives up and goes into passive coping. Now, the Dijsselhoff lab also expressed nuclear localized GCAP into virtually every neuron in the brain. There's about 100,000, and the yields are regularly between 10,000 and 60,000 per animal throughout the duration of this experiment. And so these are not freely wiggling fish at the moment, but those experiments are kind of underway. So they're fairly confident that they're able to see what they're able to see as ROIs or individual neurons. Okay. So then came a bit of clever data analysis, for which they actually didn't need theorists at all. And there were two features that jumped out of those data. And those involved specific pre-identified nuclei that people have been looking at for a long time. So habenular hyperactivation, this area called the habenula, is supposed to be hyperactive in conditions like depression. So when Aaron looked at that, he found that as a function of time, both nuclei of the habenula did make this ramp. The dashed line here tells you the onset time point of the first shock. In blue is shocked fish, and in black is control fish, again averaged over 5. And this is the average delta F over F. And so you can see this kind of ramp. Now, concomitantly with this ramp is a depression in the activity of the raphe, which is a known downstream nucleus of the habenula. So these are these two nuclei that people knew where to look at, so they looked there. And indeed, they found something consistent. So then as theorists, Matt and I said, well, what if we made up a whole story, right? Like, let's say we wrote down a normative mechanistic model from scratch. So here's how the model would go, right? The vanilla is a quencher. It says bad stuff's happening, bad stuff's happening, bad stuff's happening. Every time there's a shock. It projects to the raphe, which goes, I'm going to dump serotonin into the system. I hope you feel better. Now, if you didn't feel better, what people know from the mammalian literature is that there's an infralimbic cortex, which is part of the PFC, which volitionally and actively shuts down movement so your body doesn't go into shock. So we wanted to really ask if there's another area in the larval zebrafish that is the equivalent of the fish world's infralimbic cortex. Now, as I've set it up in the beginning of the talk, you probably can guess which way this answer is going to go. But in order to get at stuff like this, right, to test if effectively there's this almost a control theory kind of framework, a normative model that we've written down in the fish, we built data-constrained models. And since we have access to all of these data, we said, well, what if we did the inelegant thing and we just built a model that mimicked that kind of time series? And so the way we do it is, so it's not the only approach in town, but rather than making up a normative or idealized data set or training with backprop, we train these models to match the time series collected experimentally. We do this across various nervous systems, but today I'm only going to show you the larval zebrafish one. We also validate models like this with ground truth models, and then we can get into a debate at the end about how much you can trust models like this. Once we build them, we analyze them using some new methods, such as one I'm going to tell you about today, as well as some of the same methods that are used for experimental data analysis. And the goal is to infer mechanisms that are consistent to various sources of noise and implementation so that you can actually trust those values or could provide interpretability to human eyes. So how do we do this? So in order to do this to build this kind of model, and there's like just two methodological slides. So we've been building recurrent neural network models because it was one differential equation and we thought it would be a good place to start. So we take as many single module RNNs as there are regions in the brain of the fish, and we wire them together to make ourselves a multi-region RNN. And so why this becomes relevant is because the properties of models like this are given by essentially the input that each neuron gets. And that becomes key because inputs are really the why and the how a neuron spikes or fires, right? The output, what you measure, is just what the neuron is doing. So the inputs to each neuron in a model like this is everybody else's firing rate weighted by how strongly you're coupled to me. And that intuition is the current that drives your actual firing rate. So then, because of the simplicity of these models, we thought it would be easier once we build them to take them apart because this is all we've got to play with. So then the first thing we do is to wire many of them together. The second thing we do is to train the activity of each unit in an artificial model like this to match the time series collected experimentally. So in the fish case, it would be that every neuron has a target function that came from data. Not how real neurons learn, but here's a trick we use. So this is called recursive least squares. Again, not the only game in town, but if we're doing it right, it should be implementation proof. And so here's the activity of each unit. If there's no structure in the model, it will make some kind of randomish wiggle that looks like the thing in red. But at every time point, we minimize this kind of learning error until the entire matrix of connectivity is changed at every time step until the two line up. So it's a highly supervised, completely artificial trick to do this. Okay, so we've done a bunch of papers on this, but really, what do we gain from building these models? Are we really building a model that's as complex as the data we're trying to study? And so I want to convince you that that's not really true, because what we get from models like this is models that once you finish training them, can produce the dynamics that they've been trained to match. Now, depending on your taste in problems, this is either a small-sized whoop or a medium-sized whoop, right? Because you're training it to match the data, so of course it's making the data. But really, it's more than the covariance. So for people that have appreciated how hard the problem is, You can build extraordinarily large models, but you don't just get the on-manifold directions, right? Since you're fitting every neuron, you also get the off-manifold directions correctly. So that has implications. But really, what models like this buy you is the ability to look under the hood and extract from it a new connectivity matrix, which we're, to keep things general, calling directed interactions. Now, if you look in the same slide that I showed you, the method slide before, the reason this is relevant is because for a single module RNN, this matrix is predictive of how the network is going to behave as a whole. And so this one mathematical object contains a pairwise direction, type, and magnitude of interaction between all of you and me, and vice versa. And so if we built the multi-region RNN cleverly by insisting that the inter-area projections are sparse, for example, you would get a structure that is still a matrix, but now can be decomposed into these submatrices, where you would have within area connectivity in these blocks going down the principal diagonal, and a cross-area connectivity that is in these other boxes, right? And this intuition is what helps us analyze these models rather than just plotting histograms. So then these types of, so the claim I'm making is that these multi-region type of RNNs compare or are able to infer for us within and inter-area interactions that I'm claiming are inaccessible from just measurements. Please. Yes.
So firstly, so how many regions are you modeling? I mean,
so the least elegant version, 13. Because that's how many that they said would be in the atlas of the larval zebrafish. But when we do the fitting, we don't actually tell the model how many regions to expect. It gives us X number of modules. And I'll tell you what that is. But 13.
So you fit it to one data from one fish?
Yeah. Again, so another thing that I will tell you about later. But yes, each model is fit to data from one fish. But in extensions, we want to do a more generative case. But really, the thing I want to tell you about, okay, so then the properties of matrices like this are basically fascinating to me and probably Srini, and that's about it. So I'll spare you. But really, the product of these two objects, right, the dot product of these two objects gives you the thing I want to tell you about today, which is within and between area currents. And that's literally just by looking at the dot product of different submatrices with either the activity you recorded experimentally or reconstructed from the model. And these two quantities are intractable experimentally. They are verifiable with experiments, but they're not easy to get at by pure measurements alone, because they're the why and the how a neuronal output is collected. The output is the what you collect. So what does this look like, right? So let me just show you what this looks like on a ground truth data set, and then I will show you what happens when you apply it to the larval zebrafish system. So let's say we have a ground truth data set that came from idealizing this kind of fake animal that had 3 regions. And what Matt has done here is keep the inter-area connectivity sparse, and he picked the parameters of this toy model so our eyeballs have something to look at. So here's one neuron from each of these regions. Region A in blue, RNN A, region B in yellow, RNN B, and region C in red, RNN C. And imagine that the parameters are such that one neuron in region A makes something like a fixed point. Imagine that region 2 is making something like a temporal sequence, and so one neuron in it could be making a temporally sparse bump. Imagine that region C is making a fixed point that transitions to another fixed point, right? These are the outputs that you have collected experimentally. What this technique allows us to do is to now take a completely area agnostic RNN. In this case, we're picking one that has the same number of neurons as the toy model, and we fit it. Now, take my word for it, this because they're universal function approximators, kind of, this works. But after fitting, you get from this fit this kind of matrix that I had alluded to before. Now, if you do a dot product of this matrix, you're able to now decompose the recorded output in region A to 3 component inputs or source currents. And so you can see an A to A or within A current that looks like a flipped version of the output, so not that surprising. But you get 2 other components. You get a B to A current, which is reminiscent of the output recorded in region A, but is invisible in the output only view of region A. right? And then you also get a second component, C to A current, which has a fixed point transitioning to another fixed point at the same point as the recorded output in C, but is absent in the output view of region A. The sum of these three is still trivially the output that you collected, right? But if you didn't fit the matrix, if you didn't fit the RNN, you wouldn't get this matrix. If you didn't have the matrix, you couldn't do the decomposition. And so this kind of decomposition, we need to call it something, so we called it current-based decomposition, or current for short. But the fitting of the ANN really gives you the way or the units in which to decompose generated activity from inherited activity, even in one region. So what we want to do with this method is to figure out whether a recording is from a single area or multiple areas, whether there's a dynamical difference between areas, whether there's structural difference between areas, and whether there's difference in the types of inputs different areas receive. And area, the word area can be replaced with recordings or sessions also. And so the code is available online. We've applied it to many different data sets and so forth. But really, it's for in vivo tract tracing. So really, one of the questions that I'm deeply curious about, and Matt is also in his lab, is if we did do this exercise on whole brain data from other systems, smaller nervous systems, well, the fly is roughly the same size, but the worm, then without any loss from sampling, what does that connectome look like? And why does it not look like the structural connectome that has been meticulously measured? So let me show you what happens when you apply this to the larval zebrafish system. We're going back to the same data set, but remember the quest, right? We wrote down a normative thing saying we're looking for an infralimbic cortex. So I'm going to show you 3 regions, but then depending on how I do on time, I'll show you the whole brain thing also. So we build a multi-region RNN, exactly like I told you before, except because of the quest for the infralimbic cortex, we attach a 3rd region in yellow here, called the telencephalon, because we're looking for the PFC in fish for the first time, so of course we're going to look at the telencephalon. So we attach all 3 of these RNNs together, and we fit a model to it. And for those of you who are curious, this is where these 3 regions lie in the fish, and this is the actual picture. Now, this exercise works really well in terms of fitting the neural activity. And so here are 3 neurons, a habenula-like neuron in blue, Raphe-like neuron in red, and encephalon-like neuron in yellow. In gray, these 3 is the data that was used to train each of these modules in the first place. Now, to convince you that I'm not cherry-picking neurons here, which of course I am, but this is what the state space of these look like. So we're not getting every little bump and wiggle, which is probably relevant to the animal in making these decisions. But since we were after the long time scale of the active and passive coping transition, we said this is sufficient, right? We're not getting all of the PCs, we're still overfitting, but we're not getting all of the little bumps and wiggles. Okay, so the fitting by itself, as I told you, small-sized whoop. But really, because we can do this curved exercise, we're able to decompose activity in the habenula. Remember, we saw these ramps in the habenula. That activity can be now decomposed into 3 source currents. There's a within habenula current. In this case, it happened to have 2,200 neurons, a Raphe to habenula current, and a telencephalon to habenula current. And you get 1 inter-area current and 1 within area current and 2 inter-area currents just by decomposing it using the matrix extracted from the model that has been fit to data. Now, because we've taken 1 time series and we really decomposed it into 3, we can now do PCA or any kind of dimensionality reduction in the space of the currents or in the space of the input that are driving the outputs in the first place. And that brings you this kind of picture. So all this is the first principal component of each of these 3 n by time matrices. And they come orthogonalized because they are multiplying non-overlapping submatrices there, but you can orthogonalize them explicitly if you want. So you get 3 axes, habenula to habenula 1 in blue, Raphe to habenula in red, and telencephalon to habenula in yellow. Now on top, Matt projected the output recorded experimentally in the same habenula neurons and superimposed on it the times at which shocks were given to the animal. And we were hoping to see something within the habenula, but instead, you see that the early part is clustered entirely in the Raphe to habenular subspace, and it's only afterwards that the other subspaces even come up. right? And so this was sort of kind of a weird observation for us, and we didn't know how much we could trust it. But I was also raised never by Eve, incidentally, not to trust 3D plots because you can make anything look like anything else. This is what the time courses look like. This is the average time course of these currents as a function of time for control fish. So these are fish that are just head fixed, tails wiggling away, they're doing whatever they want. In black is the Gaussian convolved tail wiggles of control fish. So they're spontaneously behaving, no shocks. And the three currents are averaged across network implementations based on data from different individuals, 5 of them. And so they're doing whatever they're doing. But really, when you look at the average time course of these currents in sharkfish, you can see that the case is different. Like the early part, in which you see an elevation in the tail movements or an increase in tail movements, is actually given by rotation of a ramp in the rapid to habenular subspace. And it's only after this fish has lapsed into passivity that the telencephalon to habenula and habenula to habenula currents even come on. And so this was, and so there were two things here, right? One is that we were hoping to see something within the habenula. In fact, we wrote a cell paper on the thought that there were stronger synapses in the habenula. Now we're saying really not so much with that. It's mostly external to the rapid to habenula, at least the active coping part. Now to really convince you that we don't have to actually retract the 2019 paper is that there may be multiple things going on at the same time. And so here is what the matrix looks like. So here, what Matt has done is taken just the histogram and taken a log of the y-axis because the scales were wildly off. You also normalize these histograms so your eye has something to look at. And so in black is the baseline period in the experience of the fish. In red is the period early part of the shots, right? The active coping phase. So the active coping doesn't seem to cause any shifts in the weight distribution itself. But it does cause rotation to the current space. But it's only after the fish that has lapsed into passivity to see a broadening of the weight distribution. So what I'm telling you really, okay, so this is just a plug for curves to show you that it is actually a viable alternative to the regular way of looking at neural data, which is to look in state space. But this is just the output projected into the PCs. But here, I argue that it is a little more interpretable because the axes come with labels, because you know which current is activating when, rather than looking at something smooth in PC and making a story. So this is a conclusion from the first part. But I want to show you, and when I say second part, don't freak out. It's literally in 2 slides. So now we ask, let's go one step further, right? We've done the inelegant. First of all, we did not find anything that looked like an infralimbic cortex that went on and shut down passivity. Everything was kind of squishy. But not everywhere, right? There's separate time courses and different time scales. Well, what are the other regions in the fish do? So let's toss the shock aside for a second, and let's do the really brute force, because we can. So Matt and Manuel trained a whole brain, 13 regions, around 10 to 60,000 neurons data set to look like a single RNA, right? And so this is the same exact learning algorithm. But because we have very long time data, they also chunk the data into epochs, and each RNN is initialized to the RNN trained in the previous epoch, just to give you continuity. And this exercise, that's sort of what that is, right? We train the network and we've chunked it over time. Now, rather than getting a single matrix or multiple submatrices, you get multiple submatrices that are source and target, but they're also stacked over time. So instead of getting a curved matrix, you get a tensor of these types of curves. Now, to make sense of this, Matt did tensor decomposition to extract low-dimensional low-weights. And this is what they look like. You get a set of target weights, you get a set of source weights, and you get a time course. And because we needed to call it something, we're calling them compositional nodes. Now, because we have all these weights, and we know where the neurons lie in the brain of the fish, because we have an atlas, we can take these weights and project it back into the atlas of the fish to say, well, how many regions contributed to what fraction of a certain observed current pattern? And that's what these types of maps show you. Now, how do you go from a bulk, kind of admittedly inelegant approach to something interpretable? So this is what these nodes look like for a single fish. Now, keep in mind, there's no task, right? This is just a fish that's, I don't know, thinking about getting away probably. You do get these kinds of modes. If you ask for more modes, the method spits out multiplications or multiples of the same ones. But you really get a ramp-like mode with similar spatial patterns of neuronal engagement brain-wide from all of these fish. Now, there could be something clock-like marking the progress of time because we've looked at statistical controls, we've done controls for drift in the signal and yada yada. But you get a ramp-like mode from all fish. That's not all you get. You get a bump-like mode, you get a fast decaying kind of mode, and you get something that looks like what we're making up these names, by the way, this kind of slow decaying mode. So those of you who've seen Laura Driscoll's beautiful paper on assembling these Lego blocks, we're thinking these are a similar concept, except in the units of colors. If I want to call them compositional, they have to match these criteria, right? COSYNE people have taught me this, is that they have to be decomposable and recomposable. And if you change one, that's the only one that needs to have been changed, or it's not compositional anymore. But anyway, this kind of consistency in these modes across data from different individuals, and the RNNs are fit for different things also, is why we think we're not insane. So there's also these residual modes that are unique to each fish. So between your brain and mine, there's a common piece, and there's a piece that is completely unique. And this is a nod to Adam's talk. And there is a one that is specific to individuals, and that seems to capture a varying amount of variance of the total activity, but really is lined up closely with whole brain activation than anything else we could think of. Okay, so, and then we did statistics on it, but really I'm gonna leave you with human interpretable stuff, and then I'll stop. So really, behaviors are modulated by these squishy things, right? Internal states, environmental inputs, behavioral drive, and so forth. So one way of looking at it is to go from behavior first, right? You can cluster behavior into modules and say every time the fish makes a leftward turn, I'm going to call it syllable one. Every time it does this one other stereotypical thing, another thing. Now, this is one picture of neural modularity, and this is true of subcortical areas such as Anne Kennedy's beautiful work on hypothalamus, Catherine Dulack's work. There could be switch-like mechanisms, right? Every time switch A is on, or one region is on, it does this one behavior. And this is what we thought Habenula was doing. Habenula goes on, the fish goes into passive coping glamour. Instead, we're seeing the story as something more like this, that there are these compositional modes which are really trajectories in the state space of currents. And so if you plot the state space rather than in outputs, but in source currents, there could be a trajectory that mediates behavior one, another trajectory in the current space for behavior two, and transitions between these trajectories toggle different behavioral states. So we're viewing this as an alternative. Here's a second piece of human interpretability that I think. I think of there being a relationship between consistency and interpretability. Like if I build a model and Srini builds a model and you can't compare them on the same units, then we can't trust either one of us, right? And so this is an intuition that's borrowed from a beautiful paper, doesn't get as much traction because it's written sort of in a philosophical-ish style, Yemins and Cow. And this is the dispersion of the solution spaces of ensembles of models, right? If you thought it was wishy-washy until now, wait until I go through this. And on the x-axis is a number of simultaneous constraints. What we thought was happening, sorry, this should say fish, not one. So what we thought we were telling experimentalists to do was to get us better constraints. So we said kind of go record everything because we didn't think of anything better to say. So then they went and recorded everything, which is why we end up in the position on the top left, where all of these are valid simulations of the fish or the worm or whatever small nervous system you're looking at. But then they have nothing to do with each other because the landscape in which these models lie cannot be compared directly. So it's the multiple realization problem that Eve had written about 20 years ago that we've kind of realized. Okay, so then you get, so what we really think we want this the scenario on the right, where there's this very smooth surface, admittedly overfit, but they're completely identical, right? Like you can kind of compare one model to the other and you can extract these common features. And so rather than just simultaneous number of constraints, What we're trying to put forth is that it has to have simultaneous constraints across scales or abstractions, which is what Srini talked about in his talk and Vivek too. That if you have behavioral time theories as well as neural dynamics, or constraints from connectivity, behavior, and neural dynamics, you might be in a situation where at least the surface on which models lie are smooth enough so that the distances make sense, that comparisons can be made across implementations, even if they're not identically the same implementation each time. So consistency gives us reason to trust the models. And I think from there to interpretability could be like a straight line. I'm finished. That was just conclusion slides. I wanted to thank Matt and Manuel for their work. You, Duan, is a new graduate student who's going to take over this project and work, and then funding sources for their faith in our ideas. Thank you.